inhaled mRNA therapy for lung disorders. Messenger RNA, also known as mRNA, is a family of molecules which conveys genetic information from DNA to the ribosome in the form of a series of 3 base code words. It originates from the nucleus and is created during transcription in our body cells and carries a genetic message from the DNA to the protein-making machinery of the cell. As of recently, mRNA has also been perceived as a potential therapeutic treatment for various lung diseases, including pneumonia, constructive obstructive pulmonary disease, and cystic fibrosis. However, the greatest impediment that hindered scientists from achieving this was to discover a safe and ergonomic system of delivering mRNA molecules to the target cells. This may be possible through scientific researchers from MIT. In a paper published in POS1 called Inhaled Nanoformulated mRNA Polyplexes for Protein Production in Lung Epithelium, it is described how MIT researchers have designed a non-invasive aerosol inhalable form of administering mRNA directly to the lungs. This technology wields the potential to open up new possibilities for new treatments for lung diseases, as it is a desirable route for nucleic acid-based therapeutics. Through the research conducted, it was perceived that through the inhalation of mRNA, it was possible to induce lung cells in mice to produce a target protein, functional biomolecules addressed and controlled by biologically active compounds. The therapeutic treatment, is instigated by the messenger RNA encoding generic instructions that stimulate cells to produce specific proteins. The researchers packaged the mRNA with the degradable polymer to trick the mice's lung cells into accepting the package. Once it had been absorbed into the lung epithelium, the polymer began to grow, indicating that inhaling mRNA is an effective way to initiate the production of new and novel proteins. The scientists also repeated the experiment in genetically engineered mice, whose cells turned permanently red if they received a copy of mRNA, making it possible to count the proportion of cells affected by a dose. The researchers on this project say that if the same success rate can be achieved with therapeutic proteins, it could potentially be enough to treat various lung diseases. As mRNA can easily be broken down in the body, scientists have worked on a protective carrier in which to transport the mRNA throughout the body. Although there have been previously designed materials, many existing drugs for lung disease have previously been formulated so it could be inhaled. And an adaptation of the nebulizer was thus found to be the most efficient way, as powdered particles of medication can travel directly to the lungs. However, it was demonstrated that the nanoparticles could be freeze-dried into a powder. Thus, it may be possible to utilize an inhaler instead of a nebulizer as a mode of transporting the mRNA into the lungs, providing a more convenient means of digesting the medicine for patients, especially with those with cystic fibrosis. Different materials were tested to stabilize the RNA throughout the process of aerosol delivery. Materials including polyethylene, also known as PEI, were utilized to test the quality of delivering inhalable DNA to the lungs. Unfortunately, PEI did not break down effortlessly, and thus with repeated dosing required for mRNA therapies, this polymer was deemed unsafe as due to the potential of accumulation, leading to various side effects. Thus, researchers have turned to a type of positively charged polymers called hyperbranched poly beta amino esters, which, unlike PEI, are biodegradable and enable nanoformulation of a stable and non-concentrated polyplexes suitable for inhalation. Particles consisted of spheres of about 150 nanometers in diameter, combined with a mixture of a polymer and mRNA molecules that encoded luciferase proteins, which were suspected in droplets. The researchers have found that 24 hours after the mice tested throughout the experiment inhaled the particles containing the mRNA, lung cells were producing the mRNA protein. Furthermore, using an AI-14 reported mouse model, it was identified that 24.6% of the total lung epithelial cell population is transferred under a single dosage. The amount of protein gradually decreased over time as the mRNA was cleared. Through a repetition of dosage, researchers were able to maintain steady levels of protein, which would be necessary if adapted to treat chronic lung diseases. Further analysis of the lung revealed that mRNA was evenly distributed through the 5 lobes of the lungs and was absorbed predominantly by epithelial lung cells, which line the lung surface. These cells are implicated in cystic fibrosis as well as other lung diseases such as respiratory distress syndrome, which is caused by a deficiency in surfactant protein. If the researchers succeeded making it through the clinical trial, currently being funded by Translate Bio, a leading mRNA therapeutics company, and succeed on the medication on patients with cystic fibrosis, a repetitive dosing of the long-tailed HPBAE mRNA will generate consistent protein production in the lung without local or system toxicity. Nebulized delivery of the mRNA may provide a clinically relevant delivery system to the lung epithelium. Translate Bio are anticipating interim data in the second half of 2019 and conducting additional preclinical studies needed to proceed with the clinical trials. The fourth quarter 2018 initiated dosing of PASAGE in multiple ascending dose portion of phase 1 and 2 clinical trial for the treatment of cystic fibrosis. However, in January 2019, the company announced that the FDA placed this clinical trial on hold, requiring additional preclinical data before moving forward. Following the fourth quarter of 2019, they will resume clinical trialing with the necessary clinical data. The estimated primary completion date is dated at approximately January 2021. If this technology manages to surpass clinical trial, this could potentially provide extreme relief for those suffering with cystic fibrosis, along with other lung diseases. The treatment will change millions of lives with people who live every day with lung disease, especially those suffering from cystic fibrosis, and would become a major milestone in scientific medicine.